to the organizers for giving me the opportunity to speak here today. And thanks to all of you for sticking around. I know it's been a long conference, but I'm glad you're here. So today, first, my name's Chris Poirel. I'm a member of T.M. Morali's research lab at Virginia Tech. And today, I'm going to talk to you about some of my PhD research on developing top-down network analysis to identify and propose extensions to bottom-up physiological processes. So I borrowed this image from a nice paper by Eidinger and Laufenberger in 2003, where they talk about how computational systems biology, sort of the models that are used, span a wide range. And on either end of this range are sort of well-defined models. In particular, on the left side of this, we have what we think of as top-down models, which typically operate on whole proteome or whole genome scale data, much like the network-based algorithms that Ernest Frenkel was talking about earlier today. On the other side, we have what we think of as bottom-up models, where these models typically start with just a few interactions or a few proteins and try to explain the regulatory events among that small set of proteins. Now, typically, the models on this end of the spectrum are an order of magnitude or more smaller than the models that are on this end of the spectrum. However, the sort of ODE-based models that you get over here can often provide a much more detailed mechanistic understanding of particular biological processes. To a large extent so far, these two methods are pretty much developed independently of one another. You know, the top-down network analysis guys develop their algorithms, and the bottom-up differential equation guys develop their models, and they sort of use each other to reinforce one another, but they're not actively using each other to build better models. So in this work, we're interested in bridging the gap between these two by developing top-down methodologies that can propose extensions to these bottom-up ODE-based models. So the bottom-up model that we're going to be studying, that we're using in this work, is the model of the yeast cell cycle developed by John Tyson's lab in the Department of Biological Sciences at Virginia Tech. I'll refer to this as the Chin 2004 model, or just Tyson's model. So what I'm showing you here is just the wiring diagram for this model. It gives you an idea of the proteins or the protein complexes that are incorporated in the model, and the interactions between any pair of nodes that you see here sort of represent the reactions that are modeled by this system. Now, underneath all of this are the collection of ordinary differential equations that explain how all of these proteins change in concentration as the time progresses through the cell cycle. So this is a very powerful model. It's able to accurately predict the behavior of hundreds of phenotypes, single and double deletion mutants, knockdowns, and things like this. However, there are a handful of mutants that the model still can't accurately explain, so there's always room for improvement. Typically, when the modelers are trying to, so we've worked sort of together with these modelers a lot, and we ask them, you know, well, how do you go about trying to extend this model? And they say, well, we find a protein of interest that we'd like to incorporate in the model, and we sort of scan the literature for all of the relevant interactions between that protein and other cell cycle related proteins. Even incorporating a single protein in this model and getting it right and being able to accurately predict all of the phenotypes they've been able to so far, and then some new ones, can be a multi-year undertaking from concept to finish and publication. So in this work, we think that we can, using large yeast interactomes, protein interaction networks, we can computationally identify potential meaningful paths that will connect some of these query proteins to the known proteins in this yeast cell cycle model. And hopefully these paths can suggest extensions and improve the efficacy of these models. So here, just as a cartoon, I've got this network sort of represents the protein interaction network. I didn't want to show you one of those hairballs that sort of you can't really learn anything from. But and then here, what we've done is mark the proteins that are in, for instance, the cell cycle model. And then this would represent this query that we want to include in the model, but we're not sure exactly how it connects back to the cell cycle. So we came up with a few guiding principles for this algorithm when we first started to develop it. First, the method should be applicable to directed networks. This is primarily because a lot of the interactions that we work with are directed in nature, transcription factor events, phosphorylation events. Next, we also sought a formulation of the algorithm that's amenable to polynomial time algorithms. Primarily, this is because we wanted to be able to sort of service these queries on demand and be able to compute these very efficiently and quickly. Lastly, we wanted the algorithm to support an easy-to-interpret parameter such that when we increase this parameter, we get increasingly larger solutions. Now, this was driven almost entirely by our collaborations with the modelers themselves. We had some preliminary results where we would find links from the query protein to the target proteins. And they would look at these interactions and they say, okay, yeah, some of these are interesting, some of these are not interesting, but what we really want is more. Can you give us 3 or 4 more interactions or 10 more interactions? So we explicitly wanted to incorporate this in our method. With these ideas in mind, we developed a method which we call linker. Linker works in 2 stages. In the first stage, we first identify the relevance of each protein in the network to the given query. And in the second stage, we use these relevance scores to find highly relevant paths that link that query back to the target nodes, or the cell cycle nodes in this case. So now I'll just quickly walk you through, I'm not going to go into the mathematics behind the algorithms, but I'll just quickly walk you through sort of how this method proceeds. So here's the setup. We've got 3 inputs to the method. We've got the interaction network, the set of target nodes, and a single source protein. This is the query we'd like to include in the model. In the first stage, we identify the relevance of each protein to the query. So we start by marking that query node. And then using a random walk-based approach, we distribute this, propagate information about this node throughout the network. This is well known in the literature as PageRank. It's a random walk with restarts. So you may have heard of this before. But essentially what we get at the end is a score for every protein in the network such that it's a probability distribution across all the nodes of the network. So each value is a probability that if that probability is higher, it means it's more likely it's relevant to the, or at least proximal to the query node in the network. So once we have these probabilities in hand, we then seek highly probable paths that link the query to any of the target proteins, where we define the probability of a path as the product of the probabilities along the nodes in the path. So here you can see the most probable path from this query to any of these 2 target proteins is through this node. So next, we also introduce a parameter k, such that as we increase this parameter, we can ask for not just the most probable path, but what's the second most probable path, 3rd and 4th and 10th most probable paths. In this way, the modelers can gradually move the sliding bar and sort of see more and more interactions. So lastly, after we've identified the k most probable paths, we take this subnetwork and we identify it and investigate it to see if the interactions in this network and the nodes that are, these internal nodes that are found in the network, can you give us some clues as to how this query protein should be incorporated back into the model. Okay, so now we'll get into the data that we use. So we built a comprehensive yeast interactome by integrating a number of public databases. We used the BioGrid database, the kinase interaction database, YeastRack, and some experimental papers, this one by Bottomup et al., which gives us some phosphorylation information. Ultimately, we get a network that contains a number of interactions that include physical interactions, transcription regulatory events, as well as phosphorylation events. And we directed these interactions in an appropriate way. For instance, the transcription regulatory events are directed from the transcription factor toward the gene that it regulates. Ultimately, we get a network with 6,500 nodes and about 152,000 directed edges. After we've computed this network, we use a probabilistic weighting scheme to get edge confidence scores for this. I'm not going to go into the details of this, but it's a pretty straightforward approach. Basically, the higher the probability on this edge, the more we believe it's a true interaction. For the remaining inputs, as the target nodes, we use the 27 target proteins that are in the Chen 2004 model. And for the source, we use 12 different yeast proteins that were shortlisted to us by Tyson's lab. So these are proteins that are known to have some involvement in the cell cycle based on the literature, and also that since the publication of the 2004 model, they've investigated for inclusion into future versions of their model. So I'm not going to talk about all 12 of these today, but I'm going to just focus right now on some of the successes we've had with the query for CDC5. So what I'm showing you here is the network that we get from running linker on this interactome and asking for the k equal to 15 most probable paths that link CDC5 to any of these target cell cycle proteins. So what you can see here is, in the solid line, using the solid edges, I'm showing you the top 10 most probable paths connecting these. And the dashed edges represent the 11th through 15th pathways. So you get sort of an idea that the modeler can sort of actively scale this parameter and get more or fewer interactions as they desire. So the reason why we picked this CDC5 as a good sort of test case is that since the 2004 version, the modelers have implemented a new version of the model that has CDC5 in it. So they have a good understanding of how this connects to a lot of these proteins already. So it's sort of a sanity check that we find meaningful interactions. But also, we'd like to see if we could find something new. So on the first part of that, we do recover a number of known regulatory mechanisms for CDC5. CDC5's connections with BUB2, LTE1, and CDC14 and 15 recover its known role in the mitotic exit network. CDC5's links to NET1 and ESP1 recover its known role in the fear pathway. This is really important for the release of CDC14 during early anaphase. And CDC5's link to CDH1 plays a well-known role in mitotic exit and reestablishing the cells at the end of mitosis into G1 as they transition to a mother and daughter cell. So this is great. This is sort of reconfirming that at least the connections that we're finding are relevant. But what we're really interested in are the unexpected interactions that come from this. So there's one interaction in particular that the modelers found very interesting, which is this one between CDC5 and TIM1. So we present this network to Tyson's lab using a nice interface on the web where they can actually get a lot of information that you can't really see here. For instance, they could go in and click this edge and ask, well, why did you include this edge in the network in the first place? What's the experimental evidence that supports this edge? Right? And when they did that, they came across this paper by Rowe, Song, and Lee, published in 2002, that after further investigating this paper, this link actually explains a long-standing problem with the present model. But by that, I mean the version of the model since 2004 that they've developed that includes CDC5. I'll talk about that problem in just a few slides. So in a version of the model since the 2004 version, they've added CDC5 and its regulatory control over DBF2. So DBF2 is important in the mitotic exit network. It forms a complex with MOB1 and is extremely important in the mitotic exit network, but I'm not going to discuss any details about sort of the downstream effects here. What I really want to talk about is just the regulatory control of CDC5 over DBF2. Okay? So here's a very simple diagram for how CDC5 controls DBF2 in the current versions of their model. CDC5 inhibits the formation of this BUB2 BFA1 complex, which inhibits the formation of this TIM1 CDC15 complex, which activates DBF2. So CDC5 has a positive effect an indirect positive effect on DBF2. Now, the interesting part is what happens when we look at the BUB2 CDC5 deletion mutant. So what happens is there's an experiment in the literature that reports what happens to the amount of DBF2 when we knock out CDC5 and BUB2 from the cell. So according to the model, if you were to knock these two out, these regulatory mechanisms go away. TIM1 CDC15 is free to form, and DBF2 should be active. But the problem is, experimentally, DBF2 is inactive in this double deletion mutant. However, after looking closer at this publication that supports the link between CDC5 and TIM1, I mean, even just reading the title here, you see BFA1 can regulate TIM1 functionally independently of BUB2 in the mitotic exit network. So what that says is BFA1 can inhibit the formation of this complex without necessarily needing to complex with BUB2, even though we know this is a valid inhibitory interaction. So this suggested that CDC5 should be connected to TIM1 using this secondary regulatory role, whereby CDC5 inhibits the formation of the free form of BFA1, and then it inhibits the formation of this complex. Now if we look at the double deletion mutant, we see that these regulatory mechanisms go away, but the free form of BFA1 is now available to inhibit the formation of this complex. What happens now is the model predicts that DBF2 should be downregulated, and that matches the experimental evidence. So just as a summary, I've discussed our method linker for identifying these connections from some query proteins or some target proteins in the network. We've used this method to suggest extensions to the Chen 2004 model, and we were able to discover critical connections that the modelers sort of already knew about. But by asking for more and more interactions, we were able to identify a particular interaction that solved a long-standing problem with the cell cycle model's ability to simulate this particular double deletion mutant BUB2 and CDC5. So I'd like to talk briefly about some future directions for this work. We'd like to extend this one to multiple queries. It's not often that the modelers have a single protein that they want to incorporate in the model. It's often there's a handful of proteins they might want to consider. So this is actually sort of low-hanging fruit. The problem formulation, or the computational formulation of this, can be easily extended to do this just by making that source set more than one protein. An interesting computational problem, I think, is to identify regulatory cycles rather than just sort of finding. So right now we're addressing half of a bigger problem, right? We want to find connections from a source to the targets, but what we'd actually be interested in is finding sort of the whole regulatory mechanism. Can we find connections from the source to the targets and from the cell cycle back to the source? And not just by sort of doing the problem in reverse, but actually thinking about it and getting a full cycle. And lastly, we're currently working with Jean Picud's yeast genetics lab at the Virginia Bioinformatics Institute to experimentally validate some of these model extensions, in particular this CDC5 story that we have here. And we have some others for HSL1 and HSL7 that I didn't get a chance to talk about today. So with that, I'll thank my advisor, Tia Morali, and John Tyson for all of his help with this work, and their research labs, and our funding agencies. Thanks.
We have time for a question? Anybody? Okay. Well, thank me.
Thanks.
Very much.